Okay, so welcome back to this next video in which we are discussing experimental methods for investigating exocytosis. Okay, right, so we've just discussed the clostridial neurotoxins, which are very important tools for inactivating certain snare proteins, and we've discussed where each of the clostridial neurotoxins, the many botulinum neurotoxins and the tetanus toxin, actually cleave snare proteins. Okay, right. So what we're now going to move on to is the in vitro fusion assays. Okay, now, there are two of these, however the basic idea of them is exactly the same. So I'm going to tell you about what the two separate in vitro fusion assays are, and then I'm going to tell you about what the aim of the in vitro fusion assays is, and then I'm going to tell you about how the two different techniques achieve this aim in a different way. Okay, so the two different in vitro fusion assays are the lipid mixing assays, okay, or the lipid mixing assay rather, and then there's the content mixing assay. But as I say, the actual aim of what you're trying to achieve here is exactly the same. Okay, so let me describe what the idea, what the aim of these actually is. Okay, so this is the idea that you can create liposomes. Liposomes are the starting point for all of this. So let me just explain what a liposome is. A liposome is effectively a vesicle that is not inside a cell, basically. Okay, it is a lipid bilayer bound structure, like so, that you have created artificially, but effectively it is just a vesicle. Okay, so the idea is this: you can create liposomes of two different types. Okay, so in one population of liposomes, you can put in the T-snares. Okay, we'll call these the T-snare liposomes. Okay, so the idea is you can make liposomes which will have the T-snares in. So which are the T-snares? Well, you'll have SNAP25, so I'll put this here, okay, and also syntaxin 1 here as well. So let me just put syntaxin 1 as well. So there's the HABC domain, here's its amino terminus and its C terminus. So let me just colour those in. So here is the SNAP25 with both of those QB and QC snare motifs coloured in green. Here is the QA snare motif of syntaxin 1, and here is the HABC domain of syntaxin 1. Okay, so in one population of vesicles, you put the T-snares, and of course these will form T-snare complexes, okay, providing of course the syntaxin 1 is in the open state. Okay, then what you can do is you can have another population of liposomes, like so. So here is another liposome, and in this population of liposomes you can put the V-snares. Okay, and we will call this population of liposomes the V-snare liposomes because they are the ones which have the V-snares in. Okay, so the V-snare, remember, is synaptobrevin 2. So here is its C terminus, here is its amino terminus, and then we'll show the R snare motif of synaptobrevin 2 here in blue. Okay, so let me just label these up. So this is SNAP25 here, the synaptosome-associated protein of 25 kilodaltons. This is syntaxin 1. Okay, and this is VAMP2, vesicle-associated membrane 2, or synaptobrevin 2. Right, and the idea now is, will these vesicles fuse? Okay, we want to investigate whether the snare proteins on their own is enough to fuse the two membranes together. Okay, and the answer is that yes, these two liposomes will fuse. If you put a population of T-snare liposomes and a population of V-snare liposomes into a medium, okay, so let's just say we have some sort of container here, containing some sort of medium, and we put these two populations of liposomes in here, okay, and they'll be floating around in the medium. Basically, you are going to get T-snare liposomes fusing with V-snare liposomes, okay, and understand that it's quite rare for liposomes to fuse without the snares, okay, without the involvement of the snares. Okay, so if you just got liposomes where you had no snare proteins in at all, it would be unlikely that the two liposomes would fuse because, remember, the membranes are negatively charged. Okay, so liposomes are going to repel one another electrostatically, so it's unlikely that liposomes will get close enough to each other to fuse spontaneously without the snare proteins. Okay, so this is the idea behind the entire in vitro fusion assays. Okay, we want to see whether these two populations of liposomes are going to fuse. Now, we're not going to go into the technicalities of how you actually produce liposomes like this. It can be done. Okay, the technicalities will be horrendous. I mean, just the technicalities of how to make a liposome will be horrendous, but how to get these specific proteins in and no other proteins in, you know, it's a nightmare. Okay, but it can be done. Okay, what we're going to concentrate on is how do you actually tell whether liposomes have fused, okay? And that's where these two assays are going to differ. They both have this same objective, which is that you want to see whether these two populations of liposomes have fused, but how can you actually visualize whether they have fused? Because we can't just look at these things. Okay, so we want a way of seeing whether the liposomes are fused, and this is where these two different approaches are going to use different methods to do this. Okay, so we'll start off then with the lipid mixing assay. Okay, right. Now, the lipid mixing assay is going to rely on fluorescence. So I just want to make sure that everyone is comfortable with what the concept of fluorescence is. And then we're going to need the concept of fluorescence resonance energy transfer, FRET. Okay, so I'm going to go over those concepts first, and then I'll show you how these techniques can be utilized in the lipid mixing assay. So, the concept of fluorescence. The concept of fluorescence is that certain molecules have a property, which is the property of fluorescence, okay, where they can absorb electromagnetic radiation, so photons of a certain frequency, known as the frequency of excitation. So this Greek symbol nu here, this just means frequency, and then the EX means of excitation. So they can absorb photons of a certain frequency, and this blob here, this represents the molecule, and the molecule that's capable of the fluorescence property will be called a fluorophore. So this is our fluorophore. Okay, it's just a molecule capable of fluorescence. Okay, so it can absorb photons of a certain frequency. It will gain energy from absorbing that photon, and then the idea is it can now emit this energy as another photon, but that photon that it emits will have a different frequency, and the frequency of emission is always, absolutely always, lower than the frequency of excitation. Okay, so this is nu EM, so frequency of emission. So the frequency of emission is always lower than the frequency of excitation. Okay, and that's because how high your frequency is determines how high your energy is. Okay, so basically this is equivalent to saying that the energy of the photon of emission is always going to be less than the energy of the photon of excitation, which kind of makes sense. Okay, you would hardly expect it to be higher, would you? Okay, you'd always expect it to be less, really, than how much energy you put in. Okay, so that's the property of fluorescence. Some molecules can absorb light of one colour and then start emitting light of another colour. Okay, but of course it doesn't necessarily have to be visible electromagnetic radiation. Okay, the famous examples, of course, are where you put in a non-visible form of radiation, such as ultraviolet radiation, and then you get visible radiation, usually blue light, coming out, and blue light has a lower frequency than ultraviolet radiation. Okay, so that's the concept of fluorescence, and we're going to see examples of fluorophores later on. Okay, now there is another important concept, which is the concept of fluorescence resonance energy transfer, which is a truly horrendous title for something that's actually really simple. So fluorescence resonance energy transfer, okay, which stands for, well, which is usually abbreviated down to FRET. Okay, now this is basically the idea that if you put two fluorophores in, okay, and you pick these fluorophores carefully, then you can get a very special phenomenon occurring. Okay, so if you pick another fluorophore here, okay, so this can be fluorophore 2, so I'll call this F2 here. We'll have this as F1 and this is F2, so fluorophore 1, fluorophore 2. If you pick fluorophore 2 very carefully so that its frequency of excitation, the frequency of photons that it can absorb, is the same as the frequency of photons that this one can emit, then basically what can happen is if you shine this light that will excite fluorophore 1 onto fluorophore 1, it will produce light of this frequency, the frequency of emission for fluorophore 1, and if that's now the excitation frequency for fluorophore 2, that light will get absorbed by fluorophore 2, and fluorophore 2 will then emit light of its frequency of emission. Okay, and to make this more clear, what I'm going to do is superscript that 2 and superscript these 1s. Okay, so this is effectively the frequency of excitation for fluorophore 2 there. Okay, so this is the idea that if you pick two fluorophores very carefully, basically what can happen is the energy can be resonated to the other fluorophore. That's the origin of this name here, that the energy is being transferred from fluorophore 1 to fluorophore 2, and then fluorophore 2 is emitting the energy as its frequency of emission. Okay, right, and this concept is going to be extremely important in the lipid mixing assay, because basically if you mix these two fluorophores, you are now not going to get that much light of the emission frequency of fluorophore 1 actually coming back to you. So let's say you have a medium containing these two fluorophores, you shine this colour light on, okay, electromagnetic radiation of the excitation frequency for fluorophore 1 on, what colour light are you now going to get coming back? What frequency of electromagnetic radiation are you going to get coming back? It will be this emission frequency here, the emission frequency for fluorophore 2 rather than the emission frequency for fluorophore 1. So what we say is that effectively the emission frequency of fluorophore 1 has been quenched. Okay, you're not going to get light of this, and I'm using light synonymously with electromagnetic radiation, not necessarily meaning visible light. Okay, so basically the emission frequency for fluorophore 1 is going to be quenched. You're not going to be getting much of that coming back at you because it's all going to be being absorbed by fluorophore 2s. Okay, and that's really important, that concept of not getting this light coming back, it's going to be quenched. Okay, so that's going to be really important in the lipid mixing assay. So let's now actually move on to the lipid mixing assay and how it's going to work. So basically, actually, before we do that, let me just give you two more pieces of terminology that are going to be useful. Okay, so we call fluorophore 1 here the donor, okay, because it's effectively donating its energy to fluorophore 2, and we call fluorophore 2 then the acceptor. So that's just some helpful notation there. Okay, right, rather than calling them fluorophore 1 and fluorophore 2, we call them the donor and the acceptor. Right, okay, so we are going to use two fluorophores then. Okay, and these two fluorophores are by the name of MBD. Okay, and MBD fluorophore is going to be our donor. Okay, so this is our first fluorophore. Okay, and then the second fluorophore that we're going to use is a fluorophore known as rhodamine. Okay, which is going to be our acceptor fluorophore. So this is our acceptor. And basically, the emission frequency of MBD corresponds to the excitation frequency of rhodamine. So basically, if we excite MBD with its excitation frequency here, okay, it will release radiation of its emission frequency, which can then be absorbed by rhodamine, and rhodamine will then emit radiation of an even lower emission frequency down here. Okay, so the emission frequency of MBD normally will be quenched by rhodamine. So this is the phenomenon of fluorescence resonance energy transfer that we've just discussed, now taken into a specific example here with MBD and rhodamine. Okay, so The idea then is that we can attach these fluorophores, NBD and rhodamine, onto lipid molecules. Okay, specifically what we're going to attach them onto is a lipid molecule known as phosphatidyl ethanolamine. Okay, so we are in a position to have a go at understanding what this molecule structure actually is, because we've previously discussed the structure of phosphatidic acid. So the phosphatidyl here tells us that this phospholipid is going to contain phosphatidic acid within its structure. So let's start by drawing phosphatidic acid here. Okay, so here are the long-chain carboxylic acids, here's the glycerol molecule, and here is the phosphate group coming off the alcohol group of the third carbon of the glycerol molecule. Okay, so there's the phosphate group. Here in green is the glycerol molecule, and in orange, those are the long-chain carboxylic acid molecules. So that's phosphatidic acid. And now what we're going to do is we're going to link this phosphatidic acid molecule via its phosphate group to an ethanolamine molecule. So let me just tell you what ethanolamine is. Ethanolamine is just an ethanol molecule with an amino group. Okay, so basically, let me start by drawing the amino group here. And the amino group is going to come off the opposite carbon to the one that the alcohol group comes off. Okay, so ethanol is a 2-carbon alcohol molecule. So if I draw its skeletal formula, it looks like this. Here are the 2 carbons here, this one and this one, and they'll each have 2 hydrogens coming off, but we don't show those because we're drawing a skeletal structure here. And then off one of the carbons we've got the alcohol group, that's the ethanol. Okay, and then off the other carbon we've got the amine group, and that's the amine, so ethanolamine there. Okay, so basically what we're going to do is we're going to attach the alcohol group of the ethanolamine molecule onto the phosphate group of the phosphatidic acid. So I'll draw this just as a rectangle here, and I don't know why that's come through so badly. Okay, ignore those 2 lines that's showing from the previous page. That's just in the wrong position there. It now looks like a birthday cake of some sort. Okay, right. So that's the ethanolamine molecule there. Okay, right. And now what we're going to do is we can attach onto the phosphatidyl ethanolamine, which is this entire molecule. And I should stress that phosphatidyl ethanolamine is a normal phospholipid that you find within the plasma membrane of cells. Okay, so we can use this to build liposomes, basically. It can be a component of our phospholipid bilayer of the liposomes. Okay, so what we're now going to attach onto the phosphatidyl ethanolamine is our dye molecule. Okay, so this can either be our NBD fluorophore or it can be our rhodamine fluorophore. Okay, and very conveniently, attaching the dye molecule onto the phosphatidyl ethanolamine doesn't hugely affect its fluorescence properties. Okay, so we do it in such a way that it's the means by which we've attached it on is far away from the portion that is participating in the fluorescence activity, so it doesn't affect that. Okay, right. So the idea then is that we're going to attach these dye molecules, NBD and rhodamine, onto phosphatidyl ethanolamine. Then what we're going to do is we're going to put these lipid-bound dye molecules into the membranes of one of our populations of vesicles. Specifically, we're going to put them into the V-snare vesicles. So into the V-snare vesicles, you are going to get phosphatidyl ethanolamine molecules with both rhodamine and NBD molecules attached to them in there. So NBD and rhodamine is going to go in there. Okay, now, the idea is that you put these so densely into this vesicle that the NBD and the rhodamine are close enough to participate in the fluorescence resonance energy transfer. Now, I should have stressed this more at the time. In order for this to actually occur, the 2 molecules have to be quite close together. The 2 fluorophores have to be quite close together. Okay, so what you're going to do is you're going to put these phosphatidyl ethanolamine molecules with rhodamine and NBD attached to them at such a density that they're going to end up quite close together. You're going to end up with rhodamine and NBD molecules quite close together in this membrane. So I just draw a little bit of this. Here are maybe some of the dye molecules stretching downwards like so, and of course you're also going to have them extracellularly as well. Okay. So these are the dye molecules, and they're very close together at the moment. Okay, so each MBD molecule, which we'll show in turquoise here, is not going to be too far away from a rhodamine molecule, which we'll show in red here. Okay, and this means that if I now shine electromagnetic radiation of the frequency for MBD, the excitation frequency of MBD, so I'll put this frequency excitation and then I'll put an N there for MBD. If I shine radiation of this frequency on, what's going to happen is the MBD molecules are going to get excited by that radiation. They're going to absorb it, they're going to get energy, and then they're going to start releasing radiation of their emission frequency. Okay, so you're going to release radiation of the emission frequency of MBD. That can then be absorbed by the rhodamine molecules, which are very close by, and then of course the rhodamine molecules will have gained energy, and then they're going to then emit radiation of their emission frequency. Okay, so you're now going to get light or electromagnetic radiation of the frequency corresponding to the emission for rhodamine coming back at you, rather than light of the emission frequency for MBD coming back at you. Okay, from this population of V-snare liposomes that we have here. Okay, so that's prior to fusion. Okay, so to use our fancy terminology, we would say that the MBD's fluorescence is quenched by the rhodamine, basically, by the presence of the rhodamine, which is basically just absorbing all of the radiation that the MBD emits, basically. Okay, so what we can now do is we can mix these two populations of T-snare liposomes and V-snare liposomes together, like so, and this is going to help us to detect when T-snare liposomes and V-snare liposomes fuse, because if a T-snare liposome does indeed fuse with a V-snare liposome, what's going to happen? Now you're going to end up with a structure, let me just show this, you're going to end up with a much bigger structure, like so. Okay, now remember the T-snare liposomes did not contain our phosphatidylethanolammine molecules with the dye molecules attached. Okay, so now what's going to effectively happen is all of our dye molecules attached to the phosphatidylethanolammines, okay, they're going to become much more dilute. They're going to be much more spaced out, like so. Okay, so the dye molecules will be much more spaced out, which means that now the MBD molecules are going to be further on average away from rhodamine molecules, okay, which is going to reduce the fluorescence resonance energy transfer. So if I colour in some of these as MBDs and some of them as rhodamine, so here these can be MBDs, okay, and the other ones can be rhodamines, and inconveniently the way I've just done this, I've ended up with quite a lot of rhodamines close to the MBDs, but on average now, MBDs are going to be further away from their nearest rhodamine molecule. Okay, so there is more of a chance now that when you shine, again, electromagnetic radiation of the excitation frequency for MBD on here, you will now actually get some radiation of the emission frequency for MBD coming back at you, because now rhodamines aren't close enough to absorb that anymore, and instead it will be actually making it out and coming back to us. Okay, so basically we can detect the fusion of T-snare and V-snare vesicles by the unquenching of the fluorescence signal that comes from the MBD molecules, is the idea. Okay, right. Now, what we need to do is we need to have a control. Okay, so we remember we're trying to investigate whether the snare proteins were responsible for the fusion. Okay, now, what if the snare proteins aren't needed for fusion in some cases? What if there is some snare-independent fusion? Okay, if there was snare-independent fusion between the T-snare vesicles and the V-snare vesicles, or sorry, liposomes I should call them, okay, then you would still get the signal coming. Okay, you would still see the dequenching of the MBD fluorescent properties, okay, but that wasn't due to the snare protein. So if we want to control, we need to really have a way of stopping snare-mediated fusion and seeing what the dequenching signal you get when you mix these two populations of liposomes together is, even when there's no snare-mediated fusion. And the way that you can do that is with a botulinum neurotoxin. Okay, so your control would be you do all the same protocols, exactly the same, but then the thing that you do differently is you add to the control botulinum neurotoxin D, which, remember, cuts VAMP2 or synaptobrevin 2 in its R snare motif. Okay, this will stop snare-mediated fusion, and therefore the only fusion that you're now going to get in this control is going to be snare-independent fusion. Okay, so the signal that you then get when you add these two populations of liposomes together, the dequenching signal of the MBD fluorescence that you get, that will be the signal from the snare-independent fusion. Okay, so you'd want to subtract whatever that signal is from the signal that you get due to, well, in the non-control where the snares are intact, basically. So that's your control to make sure that you can subtract off the non-snare mediated fusion, or the snare-independent fusion as it's usually called. Okay, now, there are some technicalities that I need to tell you about which might be a little bit concerning, okay? A little bit concerning about the physiological relevance of these in vitro fusion assays. Okay, so the first thing to say is that to actually get a signal that you can see, okay, to actually get a noticeable increase in the amount of emission frequency radiation that you're getting of the the MBD emission frequency, to actually get a noticeable increase in the intensity of that, what you need to do is you need to have these V-snare vesicles with very high copy numbers of VAMP2 within them. Okay, so these liposomes here, these V-snare liposomes, and I'm sorry I keep mixing up liposomes and vesicles, they are fundamentally the same concept. Okay, it's just that liposomes are a experimental construct, and vesicles, the difference really is that vesicles, of course, contain a huge number of different proteins. We've put in a few proteins into these. In reality, vesicles, synaptic vesicles, contain a huge number of other proteins that aren't in these. Okay, so that's the difference between liposomes and vesicles. So I should, strictly speaking, say liposomes, but I'm often confusing it and saying vesicles. But don't let that confuse you. Okay, so basically, where was I? Oh right, yes, copy number of VAMP2. So basically, in order to actually get a noticeable rise in the fluorescence signal that you're getting from MBD, you actually need to have really high copy numbers of VAMP2 in here. Okay, so in these liposomes, you need to have a copy number of VAMP2, which means the number of VAMP2 molecules per liposome of around 300. That's really high. Remember when we discussed synaptic vesicles, their copy number of VAMP2 was closer to 70. Okay, so maybe we're taking this to non-physiological levels here. Okay, but that's the level that you have to take it to get a physiological, well, to get a noticeable signal. Okay, the other thing, another technicality, is that you need 5 times as many T-snare liposomes as you do V-snare liposomes, again, to get a noticeable signal. Okay, right, so even though this does work, if you do this assay, you do find that the T-snare and V-snare complexes do fuse, and it's much bigger than the snare-independent fusion that you get when you do it in the presence of botulinum neurotoxin D. Okay, so it does work. These proteins on their own are powerful enough to fuse these two liposomes together. Okay, however, bear in mind that you have to take it to a 300 copy number of VAMP2 in those liposomes in order to get this fusion, okay, which is non-physiological. But at the same time, what you should also note is that we are lacking loads of regulatory proteins here. We are lacking, for instance, the MUNC18, the MUNC13, and those other active zone proteins which might be involved in getting the syntaxin 1 ready to actually form these sort of complexes. Okay, so potentially that might explain why you need such a high copy number there in order to get a signal. Okay, right. The final thing that I want to say, which is a pro for this, which is how useful it is, okay, is that what you can now do is you can also add into the medium where you're doing this in vitro fusion assay, you can add in regulatory proteins and see if they increase the signal. So what you can do is you can decrease the copy number of VAMP2s in the V-snare liposomes, okay? And then, of course, the signal that you get when you mix these two populations will be non-noticeable, okay? So you won't get a noticeable increase in the NBD emission frequency radiation coming back at you, okay? Then what you can do is you can add regulatory proteins in that you think might be involved in snare-mediated fusion of the two liposomes together, and you can see if that now produces a noticeable signal, okay, and that would then tell you that that regulatory protein was involved in helping the snare complexes to fuse the two vesicles together, or the two liposomes together. Okay, right, so that is the lipid mixing in vitro fusion assay. Okay, in the next video what we will do is we'll look at the other way that you can do in vitro fusion assays, which is the content mixing assay, which is a very, very similar concept.